Very good evening, everyone. I am Dr. M. Shreya Jaiswal, currently studying in Pusan National University, South Korea, and I am the host for today's session. Today, we have Dr. Rakshit Sethi sir with us. He has done MDS in oral pathology from Raja Rajeshwari Dental College and Hospital, Bangalore, and along with that, fellowship in forensic odontology from DY Patil, Mumbai. Moreover, he is having 15 years of experience and currently working as a professor in Kusum Devi Sundarlal Dugar Jain Dental College, Kolkata. So the topic of today's session is Ameloglyphics with colorimetry, a multidimensional tool. As you know, this topic seems to be so captivating as forensic odontology is evolving over time and has opened newer avenues that may help in identification of individuals. So before starting the session, let me tell a few points to all the attendees over here. If you have joined live stream from YouTube and Facebook and have any queries or questions, you may ask by commenting and you will get your answers at the end of the session. And if you have joined via Zoom link, you can write your questions in Q&A box, but not in chat box, okay? Q&A box in the control panel, and it will be answered accordingly. So all over to you, Dr. Rakshit sir.
Thank you, Shreya. Am I audible first and foremost?
Yes, you are.
So very good evening to everybody. And Ameloglyphics is a topic on forensic dentistry which is untouched. So can I share the screen?
Yeah, sure, sir.
Is it visible? Yeah, can you see it?
Yes, it's visible.
Right. So good evening to all those people who are here right now. I'm going to present a topic about Ameloglyphics. So what is Ameloglyphics and why is it important? So basically, forensic dentistry is about individual identification and gaining importance like in a lot of places and lot of manner. So number of methods have been used. So I'm sure all of you know what is rugoscopy. Rugoscopy is the study of the palatal grooves. Kiloscopy is the study of the pattern of lips. Bite marks, which helps in identifying, people who are under criminal records. Bite marks help. Now this tooth imprints, radiograph, photographic study, and molecular methods. These are different methods in which we have forensic dentistry as one of our main. So rugoscopy, can you see those imprints of the palate? Kiloscopy with the prints of your lips, different prints, and bite marks helping in a lot of criminal cases. So accordingly, our country is a little less, comparatively less important in forensic dentistry we give. So why enamel imprints? Out of all these various methods, ameloglyphics is the particular interest of enamel, as the enamel of teeth is highly calcified structure that resists decomposition and therefore serves as a unique tool for identification. Now, and like all of us have fingerprints, the enamel also is made up of calcified rods. So the rods are nothing but an accumulation of all the calcium hydroxide apatite crystals, which gives it a form of a prism. So this prism enamel structure can be is actually singular for a particular person. I might have one particular pattern. You people might have one particular pattern, like how we have fingerprints, and all the pattern of each tooth is going to be different. Now, this pattern, when we use it, it can also help us in identification and possible correlation between the occurrence of several congenital defects and acquired disease. So it is seen that This in particular, I'm talking about kiloscopy. They have in Kerala there was a study which was made by comparing the male kiloscopy and female kiloscopy. So when they found the lip imprints, these imprints, if they had a particular shape or structure, they could detect that the child which is going to be born could have.
Dr. Shetty, sorry to interrupt. Could you please make the PPT full screen?
It is already full screen.
Okay, okay. I'm sorry. It's not being shown as full screen.
It's not being so. That's why I messaged you, sir, that can you please do the slideshow? Because in our screen.
One. Now I went full screen. You still can't see it?
No, sir.
Yeah. Sir, can you try once by pressing F5 button?
One minute.
Yeah, sure.
Now full screen.
Yes,
Yes, sir.
yes. Thank you, sir.
Thank you. All right. So all of us know that that's what I was talking about kiloscopy. You know, the lip imprints of two people, especially the male and the female genders, when the lip imprints were taken of a married couple, so they could detect that a disease of cleft palate could be identified. It had a matching. So that is what is the importance of kiloscopy. So Ameloglyphics also helps in identification of certain diseases like enamel hypoplasia, the rods being weakened, and because of the cracks produced in the weakened teeth, we might be able to come to this. So this is a study which has to keep continuing. So the use of these patterns, most probably what we have done is, so why this enamel imprints, why is it so important? Basically, it is a product of ectoderm derived cells. The process of enamel formation is complex and organized. All of us know that enamel is laid layer by layer, and it has got a rhythmic pattern deposition of around 4 micrometers with a rhythmic pattern of deposition from 5 to 11 days. So it is carried out by specialized cells called ameloblasts. So the cell laid on enamel rods that are the basic structural unit in an undulating and intertwining pathway. This is reflected on the outer surface of enamel as pattern of ends of a series of adjacent enamel rods. So this histology, I'm very sure all of those who have done videos will understand. So because of this complex enamel structure, it is easy for us to identify the enamel imprint. So like fingerprints, Ameloglyphics is toothprint. So right now, this is how we recorded the pattern of Ameloglyphics. So we use a sellotape. This anyway I'm going to describe as we move further. So this particular pattern of the teeth could be taken on a sellotape, and this print which we are getting is what we are talking about, Ameloglyphics. So this print is going to be different for me. It is going to be different for maybe Manushya, maybe all of y'all. So this is a particular print we are talking about, or Ameloglyphics. Now, to achieve this ameloglyphics, to get these imprints, the most important part is that we need to keep the teeth dry. First and foremost, all of us know that biofilms form on the tooth as and when we stop, we eat, or we when we brush our tooth, within 10 minutes it forms. So what we did is to remove this biofilm, we made the patient undergo scaling. So once they underwent scaling, we have a window of 15 to 20 minutes where the biofilm will not form. After that, these impressions were taken. So it is always advised that you will be clear of the biofilm so that the prints can come up easily. So once you get these prints, then I will be discussing what we do. So we took a small, I just did a pilot study initially in coordination with one of our neighboring colleges. It's called Most Institute. So he helped me in getting this device, which is called as colorimetry, which detects the color of the photograph which we give. So prior to the study, there was an ethical clearance from our college institutional ethical committee. So what did I do is I took in 3 sample size, that is of 30-30 each. So I divide them into 3 to make whether we can differentiate between males and females. Because I'm right now in Kolkata, so I took Bengalis and non-Bengalis, and the group C contained people of like a father and daughter, or mother and son. This is the reason is because most of the hereditary patterns, the first child normally has all the fathers or mothers genetic components. So this was the reason I took one parent and one child. Now the material which we use is the most cheapest and the easiest material we can use. Normal your slides which are used for your histopathology, or you use slides for your normal histology. Those slides, normal sellotape, scissors, the least cost-effective material. So it is very easy for us to actually do this study. So as I told you, now to remove this biofilm, we subjected every individual, those 90 patients, whoever came, we subjected each of them to the oral profile axis of anterior teeth. I just started with the study with only doing the anterior teeth. I did not go in further for the posterior teeth, because now what I have done is I am just doing a pilot study. So it is a small study. I need to make it more valuable. The number of sample sizes have to increase. So right now, I'm just with 90 patients. I might even increase the patient to come to a proper conclusion. Anyway, we have got results in this 90 patients itself. So the individuals were subjected to localized oral prophylaxis of anterior teeth in our department of preventive healthcare dentistry. Then each of these individuals were brought to the department of our oral pathology. I am a professor in the same college. So they were seated onto the dental chair. The anterior tooth were then dried using cotton, and the impression of the enamel imprints were obtained on a cellophane tape. So I'm just showing you exactly how are we going to get this. So right, so this is the process. So first the patient is cleaned. There is an cellophane tape, small piece removed. Okay. It is attached. So it was the imprints were taken, and the imprints, after taking, it is directly attached to the slide. This is as simple as the procedure is. So once we finish this procedure, the next step what we need to do is a very simple and easy step, which nowadays all of us have, the mobile, okay, camera, just any normal camera with we just need to take a snap of this snap, and then the cellophane tape was then transferred to the slide subjected to photograph. Then this particular photograph which we take was subjected to the image analysis colorimetry. All right, so this is the next procedure. So image analysis colorimetry. This is a more clear picture of the enamel rod ends we are talking about. This pattern is particular for every individual. For me, it will be different. For another person, it will be different. So this, to come to a conclusion, we analyzed this particular photo into the image analysis software. So what that software does is it calculates the red, blue, and green wavelengths and then puts it into a particular graph. Now this graph is the graph which is going to show that this particular graphing is going to be for a particular individual. So we had two types of graphs which we got for every individual. So what is image analysis colorimetry? Let me just come into this. This is, again, I told you, it was Swarnendu Singh who actually helped me with this. So he has made a device which is called as image analysis colorimetry. So this is a technique utilized image processing of acquired images by studying pixel by pixel analysis. So the colorimetry is performed by extracting the red, blue, green value from the images, and this helps to understand the batch to batch variation and color depth information also. So this is how the technique is used. So this software is developed in lab view to platform, and there is an intuitive user interface. So basically an artificial intelligence type of software. So the number of images I keep sending, it keeps easily identifying. So more the number of images I have, the faster and the more intuitive this particular software becomes. So this data is developed, and the graph to which is corresponding after getting the red, blue, green value is put into a graph. So we get a graph like this. Now we have a red colored graph and a black colored graph. Now this was, now you can see the above part, there are two images. So image one was of one particular patient, and image two was of another particular shade. So image one was of black and image two was of red. So both these patterns was different. So this is especially for a gender differentiation. We had to verify whether it was a male or a female. So the red one showed that it was a male, and the black one showed it was a female. So we achieved this graphic image and showing two different graphs, representing two different graphs, and making it individual for a person. Now there are a lot of questions which might run into your mind, that when we take a photograph, can we change the direction of the slide and take it in a landscape? You take it however you want. We tried all this. After trying it, the image and the graph, which we tried to subject it to different type of angles also, the graph showed the same kind of graphic representation. There was no change in the graphic representation. So finally, we came to a conclusion that the gender identification and the the parent and the child identification could be done, but we were not successful in the ethnicity. We did not find any changes in the ethnicity of the person. So this was the only drawback in this particular study which we have done. Okay, I need to play this video. Will I be able to support just to show what is colorimetry image analysis? How does it study? I don't think. Okay. Can the tech team please can you play this video if possible? Can you all play it?
Sir, there may be any option in your screen only to play the video at the bottom. You can see here at the left to bottom side. I think you can get it.
Just let me one minute.
Okay.
I'm sorry, I cannot play this video. Okay, anyway, I think all of you understood with this picture itself that how this is the video which I had, how it analyzes and how the graph was going to be formed. So this picture and these values all put together gives a graph representation. Okay. So now coming to the main discussion, I exact onto why did I choose Ameloglyphics. So what is the different methodologies which were used in different studies? Maximum studies of Ameloglyphics was all done with the tooth extracted. They used to basically what was done is certain studies had selected the central region of the tooth surface. The buccal lingual surface was selected. Then 37% orthophosphoric acid was put to demineralize the enamel. Then the impression of the tooth taken by a cellophane tape. This print was transferred on a slide, and the slide was subjected to biometric analysis. The disadvantage of the studies in the other studies are that biometric analysis is a very complex method. So, and biometrics is not easily available. The biometrics are in special cases like the Central Bureau of Investigation, or you'll have to contact the police. This is a very lengthy process which has to be done. And all these tooth which they extracted, it was on an extracted tooth. So we used our mind onto why not use the same method into just putting the tooth directly from a patient. Why extract it, then do the study. So this also is one advantage we got over this. So this is the method which was previously used. How the tooth was being, how the enamel rods were being analyzed. So here, so this is the way it went. So here they put the orthophosphoric acid, then clean, dried up, then the cellophane taker, then the rods being analyzed, and then it was put into biometrics to determine the pattern of the enamel rods. So what we did was the present study. What I have done, all the 90 patients which I got in, especially the 30 males and females, and the 30 parent and the child, our study was pretty successful and we got a good result. So the present study concluded there were no common enamel rod end patterns among the study group. That is, no two rod end patterns of the carious and non-carious groups were found to be similar. So this is what they had found in their study, which group we just spoke about. So no particular pattern was found to be unique to either of the study group. Each tooth of the study group showed a different pattern. So the pattern varied in different areas of the print from the same tooth also. So in the study which this particular study which we are talking about, the study in which they have done, they could not get the same enamel rod pattern from each other. I mean, carious and non-carious, they tried to find the pattern, so they did not find any difference. But when I started with the study, so I wanted to first achieve whether we could differentiate between a male and a female gender. So that we luckily we have achieved. So this study also was not a very effective result is what they're trying to tell. So most of the studies showed that enamel pattern did not correlate much with the diseases. So I got it into forensic to get it into a study maybe to help in gender determination or any other particular studies which can be helpful. So this pilot study was done by us in a new method of study and found it to be much more reliable than most of the other studies. Now the advantage of our study is very simple: non-invasive method, cost effective, no chemicals or nothing to be used, and this is the first study done under image analysis colorimetry. No extractions, no trauma involved. Achha, now there are going to be certain disadvantages. Yes. Now, when you remove the picture which we take or the cellophane tape which we take the enamel imprint, that the problem is we cannot store that particular imprint. The imprint disappears. Right. This is the only disadvantage which we found in the. So maybe you can take multiple cellophane tape imprints and put it on a slide and take the photograph. So the photograph is the best way to maintain this particular slide to be kept for image analysis colorimetry. So the retaining of the slides with the cellophane tape is going to be a disadvantage for us. This is one which we found disadvantage. Right. So now this is a graphic representation after the image analysis colorimetry. Yes, this image analysis colorimetry, the particular software which is developed is also easily available by this institute, Bose Institute. So if anybody is really interested in some studies, you can join in to do this. So this is the image analysis colorimetry. We found 4 or 5 individuals with different graphic representation. So each graph showing a different unique variation. So this is how we came to a conclusion that this study can help us in achieving a lot of success in our future studies. So I want to even make this study bigger. So what I've come to a conclusion with this particular study was that the study proved that Ameloglyphics can also be used as an in vivo study. Everybody had to have an removal of the tooth, extract the tooth, and then study it. So what I tried to prove here was that the study does not need extraction always. We study the enamel rod ends or the enamel rods as a structure. We are getting good enough material from the enamel rods after the biofilm has been removed. The study showed that an individual identification can be done using our method. So you could identify a particular pattern. But only the thing is the pattern has a graphic representation in our study. The graph represented 2 different identities and hence proved that this method is reliable. However, as the software used for image analysis needs a larger sample size to recognize a pattern and to make the study statistically significant. 90 patients, in fact 30 was not a very big statistical significant study. But yes, we have started working on this project again and we have increased the sample size. So maybe increasing it up to 500 cases. So now once we use this, then the software will definitely have a statistical significance, which is going to be high. So I would like to thank few people. I would actually like to thank the Dentist.Online channel for giving such an opportunity. Manushya was my student in Vanachal, so I'm very proud that she's doing really good. Congratulations, Manushya. And the list of people, Dr. Ashish Acharya, I think most of us know, who was one of my main inspiration and under whom I studied forensic odontology from D.Y. Patel. And those who don't know, Ashish Acharya sir is one of those people who helped that one of those in the Nirbhaya case, he proved that one of them were juvenile. So Mr. Samendra Singh was a scientist of SN Bose National Center who actually helped me, my department, presently the HOD and principal, and my students who actually helped me do this study, who got me these, they really gave their imprints to get my study done. Thank you. If you all have anything to ask, kindly ask. I'm sorry I finished it fast because my sample was really small. So I hope this has been effective and we need to make forensic dentistry forensic odontology more reliable. Now the problem is in our day-to-day clinical practice, none of us actually just collect data. It is very important. So the importance only comes like how other countries, Western countries use is more of materials. They keep methods. They keep systematic X-rays. I request all the dentists to do this. Keep an impression material. It helps us in the future. So any kind of reference, any kind of problems, we can be of a lot of help to the society. Thank you. Over to you, Manush.
So, yeah, thank you so much, sir. This was very insightful presentation. So we will start Q&A round. Before that, let me share my screen and let me share my presentation for all the attendees. So I request you, sir, can you please stop sharing your screen? Yeah. Okay. So let me share my screen. So before the question and answer session round, so I would like to talk about the DentistChannel.Online platform, which is emerging to be the largest digital platform who supports all the dentists and the dental students in conducting the webinar or seminar, any kind of interactive sessions, any kind of related to any dentistry businesses and all. So we motivate all the students and all the dentists all over the world to come forward for sharing their valuable knowledge. So these are the services that we use to give in DentistChannel.Online. The left side is for the dental profession and the dental students, and the second one is for the dental organization. So this is the thing that this is the key highlight for the DentistChannel.Online, that is 700 plus live dental webinars we have conducted for now, and 40,000 plus participation certificate issued. Along with that, we have 25 plus dental workshops attended till date, and 400 plus national and international speakers have conducted the webinar and seminar through DentistChannel.Online. Along with that, 300 plus oral care videos are saved under the Save the Tooth. And one more, this is a proud moment for us, as we take pride in sharing the DentistChannel.Online entered into the India Book of Records for having maximum speakers participating in a virtual conference in oral implantology. And if you see that if you get the Prime membership, so these are the Prime membership benefits that you will be getting. That is, you will get the regular updates and news regarding the dentistry, and then access to the scientific articles and recorded webinars. Along with that, you can participate as a speaker, then publish your public awareness videos, and certification by recognized institution with CPD or CDE points. And then you will also get a special discount on dental courses, and you will have the access to all the webinars organized by DentistChannel.Online. Moreover, you will also get a personalized account in DentistChannel.Online with access to profile. So this is somewhat look like this is a CPD or a C point that you will be getting if you participated in this. And so don't be late, just hurry and join the membership now. And you will get a discount if you use this promo code, that is SJ100. I repeat, that is SJ100. So you will get a discount over here. And these three are the upcoming webinars of this month. The first one is the myth about dentistry and overcoming by awareness. So, and next followed by this is the upstream and downstream approach in public health, and then oral manifestation of hematological disorders on reported cases. So we also have all SNS account in all the SNS thing like Facebook, Twitter, Instagram, LinkedIn, YouTube, WhatsApp, and Telegram. So if you want to have, if you have any query, any doubt, so you can save this number in your phone, and you can WhatsApp, and this is the address for the DentistChannel.Online office. So this was all about the session today. So now I would like to again I will open the Q&A round, and for the for sir to answer the things. So first, sir, this was the very insightful presentation, as I told you before. So the next thing is that I would like to ask one question from my side, that you used cello tape, right? Cellular
Yeah.
tape you used. So there, I think I have heard that you can also use cellulose acetate, and so cellulose
Cellulose,
acetate.
yeah, that's what I'm trying to tell you. Cellulose acetate, to get it in your day-to-day activity is going to be a little difficult. Cellophane tape
Yes,
is the
yes.
easiest and the simplest method of using it. So I saw, and it didn't differ in the quality. I mean, the only disadvantage was the cellophane tape did not resist, or I mean, does not retain the pattern for a long time. Because of the sellotape getting easily shrunk and easily foldable, it was not able to retain. Otherwise, for 10 to 15 minutes, definitely you have the image. You can easily capture good images with a normal phone. So that was the cost effective. Also was one of the reason why I chose cellophane tape.
Yeah, I got it. So I think this is gonna be a reliable technique, right? Because as if any body has been decomposed, so if we have this calcified tissue, so we can identify the criminals or anything.
We have a question, I think.
Yeah. I'm saying the question is from the Preetham Viswas, that what least material do we need to collect the samples?
Least material, nothing. Your normal gloves, your normal what you use in your clinic. Just you need a cellophane tape and normal glass slides, which is easily available in any market. Especially if you go to any laboratory, these are the only materials you need to collect the sample size. So nothing much. Only a cellophane tape, slides, and your normal gloves, mouth mask, what you use in your daily chamber.
Okay, I got it. So I hope this Preetham Biswas, he might have got his answer. So I think this is gonna be an emerging thing, and I think further studies are still needed
We need a lot of larger sample size, and now it is just that I've started with only gender identification. Now I'm going to even start maybe continue the study with comparing two particular diseases. Let us start off with caries only. So maybe I will start studying the pattern of caries. This we have to do, or this maybe can be a research. So the number of research, the main thing is the research. So as many as researches we do, that much we are into advantage.
Yeah. There's one more question from Prachi Agrawal. So she is saying that, what about the software, sir? Good evening for very amazing session. And what about the software, sir?
Software is easily, that's what I'm telling you, those who need it. This SN Bose Institute from Kolkata, they have developed the software. This is basically with their help, I could achieve this particular study. So this software is very easy. And I think it is this software is cheap also. I mean, cheap in the sense the software costs a little, but I think once you have it, and if you do multiple, it's not only about this. Image analysis, colorimetry is just one small. So you can do a lot of other studies using colorimetry. Other than Ameloglyphics, a lot of other studies can be done. It is easily available. Contact, I've told you, Mr. Swarnendu Singh is the scientist who developed this. And I'm in constant touch with them. So anybody needs it, I will give you all the number, so you all can contact him and directly get it from that institute.
Okay. So, sir, there is one more question from Dr. Aditya Kashyap. How exactly will be able to differentiate using graphs? I mean, between male and female. How exactly we will be able to differentiate using graphs? I mean, between male and female.
So there were two. If you all saw, graph had two timelines. We had those markings. So the red, blue, and the green would calculate and give us a value. So every individual, the value was different. So that had a graphic representation. So my value, if I had to say, my value showed the graphic representation at around, and the longitudinal way it showed around 500, and in the cross section it showed around 550. So this graphic representation proved that this is my particular graph. So same way, the 90 people had different 90 readings, or the 90 graphical representation.
Okay. So I hope that Dr. Aditya Kashyap might have got his answer. So one more thing, sir, I would like to ask that you have divided the sample patients into 3 categories, like.
Yeah.
So I want to ask that was there any sex predilection? It means that whether a female was more or male was more, like something like that. Was there any?
No, I made it equal. 15 males, 15 females.
15. Okay, yeah, I got it.
What I tried to do was only that, I wanted to differentiate that with this basis, can we identify a male and a female gender? So that I successfully got it. Now, I did not take an age group. There are a lot of disadvantages in the study. Again, I have not taken an age group. I used all youngsters this time, all my students. So 90 students were. So now the next question will be whether I can use the study in older age, above, because, the enamel tends to reduce in its thickness as you go older. So these are the research which still has to be made. Whether the pattern remains the same or will it show any difference?
Okay. So I think only those much questions were here on the chat box. So I think it's all about today. So thank you so much again, Dentist Channel Online, for conducting this webinar, this very insightful, very hot topic, very evolving topic. And I would like to thank again Dr. Rakshit Sethi, my teacher, my first teacher. So yeah, thank you so much, sir, for such an insightful session. And I think it is going to be very helpful for all the dental students and the dentists who are working in this topic, like in forensic odontology.
Actually, those who are interested in forensic can make a lot of studies of this particular topic. It's not many studies have been done.
Exactly,
so I'm also thankful to Dentist Channel. Thank you very much. Thank you, Shreya, contacting me and giving me this platform also. Thank you so much.
Thank you so much, sir. So thank you, everyone, and thank you so much. And I would like to conclude this session. So thank you to all the attendees who attended this session, and thank you, sir.